Keeping up with our motto, let learning be a joy and teaching a pleasure. Here we are with a remote teaching and learning process to bridge the gap. Happy learning, students.
Hello students. Today we are going to see the chapter from Science and Technology part 2. Chapter number 2, that is Life Processes in Living Organisms part 1. We have covered the first and second part of the same topics. Let us recall the topics which was covered in the first and the second part. The topics which are covered are living organisms and life process, living organisms and energy production, some nutrients and energy efficiency, and cell division. These are the topics which was covered in part 1 and 2. Topics to be covered in this part is mitosis. Mitosis is a very important stage which is carried out in cell division. But before starting with the part of mitosis, let us understand about haploid cell and diploid cell. Haploid cell is basically a cell which contains a single set of chromosomes. And since it contains a single set of chromosome, it is called as an haploid cell. Whereas diploid cell is a cell which contains two sets of chromosomes. A cell which contains two sets of chromosomes is called as diploid cell. And since it contains two cells of chromosomes, it is represented by 2n. Now this haploid and diploid cell can be understood with the help of this diagram. In the first diagram, you can see a single set of chromosomes is present, so it is called as an haploid cell. Whereas in the next diagram, we can see two sets of chromosomes are present, so it is called as diploid cell. Basically, mitosis is nothing but a division of a cell. So let us understand the structure of the cell first. This diagram shows you the basic structure of the cell. In the structure of the cell, at the center of the cell, we can see a white ring is present. This ring is called as nucleolus. Inside this nucleolus, you can see black thread-like structures are present. These black thread-like structures which are present inside the nucleolus are called as chromatin. And at the top, you can see a white patch. This white patch is called as centriolus. So basically, we have seen a white ring called as nucleolus, the black thread-like structure called as chromatin, and the white patch which is called as centrioles. When we have heard about nucleolus, we have heard about nucleolus containing chromosomes. So basically, those chromosomes are formed from this thread-like structure called as chromatin. This structure of chromosomes is shown at the bottom. In this structure of the chromosomes, you can see a knot which is present at the center. This notch which is present at the center is called as centromere. A stage is reached in the cell division where this chromosome splits up. When this chromosome splits up, they are called as sister chromatids, which is shown in the diagram this way. So basically, the chromosomes is splitted into two parts, and when they split into two parts, it is called as sister chromatids. So there is a difference between chromatin and chromatids. Chromatin are the threads which are present inside the nucleolus, whereas sister chromatids are nothing but we are getting it from the chromosomes. Now, after understanding the basic structure of the cell, let us come to the part of mitosis. This mitosis is carried out in two stages. The first stage is called as karyokinesis, which is nothing but nuclear division. This karyokinesis is carried out in four phases. The first phase is called as prophase. In this diagram, you can see the same structure of the cell. Now, what happens basically in prophase is the condensation of thread-like structure starts. That is, the condensation of thread-like chromosomes starts in this phase. Due to this, they start to appear with sister chromatids. Centriole duplicates. If you recall, in the initially we have seen only one centriole. In this stage, the centriole duplicates, and each centriole start moving to the opposite poles of the cell as it is observed in the diagram. The nuclear membrane which is present, it starts to disappear by the end of this prophase. So basically, three things has been carried out. The sister chromatid has appears, the new centrioles duplicates and moves to the opposite poles, and the nucleolus starts to disappear in prophase. Now this stage is continued to the next phase called as metaphase. In metaphase, the nuclear membrane completely disappears. The nucleus membrane which was started disappearing in the prophase, here they have completely disappears. The chromosomes complete their condensations and become clearly visible. You can see it in the diagram. The chromosomes are present at the center in four different colors, and at both the two poles the centrioles are there. All the chromosomes are arranged in the equatorial plane of the cell as it is seen in the diagram. Now joining this centriole and the centromer, you can see thread-like structures are present. This special type of flexible protein fibers are called as spindle fibers, which are formed between the centromer and each chromosomes and both centrioles. So these are called as spindle fibers. So these spindle fibers are formed in this metaphase. Basically, what the spindle fibers does is they starts pulling the chromosomes and break these chromosomes into sister chromatids, which is carried out in the next phase called as anaphase. In anaphase, the centromer split and thereby sister chromatid of each chromosomes separate, and they are pulled apart in the opposite direction with the help of spindle fibers. The spindle fibers is basically pulling those chromosomes and breaking them into sister chromatids. These separated sister chromatids are called as daughter chromosomes, and each set of chromosomes reach at the two opposite poles of the cell. So that's what happens in this stage called as anaphase. Then it comes to the last phase called as a telophase. The chromosomes which has reached at the opposite poles of the cell, now start to decondense. The condensation has started in the prophase, now they are decondensing it. So they again became thread like, thin, and invisible. The nuclear membrane, which started disappearing, the same nuclear membrane is formed around each set of chromosomes. Thus, two daughter nuclei are formed. If we recall the diagram, in the prophase diagram, you have seen only one nucleus present at the center and one cell was there. But now, in that one cell, two nuclei are formed. But these nuclei are called as daughter nuclei. So in this case, the nucleus has got separated or has got splitted into two daughter nuclei, and here is what the karyokinesis stage is completed. And once the karyokinesis is completed, it moves on to the next stage called as cytokinesis. In cytokinesis, the cytoplasmic division occurs. The notch which is formed at the equatorial plane of the cell. This notch deepens gradually and two new cells are formed. In plant cell, instead of notch, cell plate is formed. These new cells are called as daughter cell. That means in the cytoplasmic division, it is getting converted into two daughter cell. Till telophase, only one cell was there and two daughter nuclei formed. In this phase, it is completely separated into two daughter cells. This is how the whole mitosis is completed. Thank you, dear students.